The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone. Thank you all for joining this webinar from either your office or at home in this situation. The title of this webinar will be the analysis of high resolution HLA typing by NGS and which tools do we need. So my name is Nina Lauterbach, and I'm one of the senior field application scientists at Omixon, and I will be the presenter of this webinar. Since it's already 3 o'clock, I will just directly start, and everybody can still join in the meantime. So to start with the agenda, I will first start with a very brief history on the molecular techniques and NGS. Then I will start on highlighting some of the main differences between Sanger sequencing versus NGS. I will then talk about the benefits and the challenges that comes with NGS-based HLA typing. And then I will talk about the software analysis and the tools that we provide in the software. I will then end with a summary, and if you have any questions, you can then add them into the chat box. So when we look back into time, the molecular testing started around the 90s, and it all started with the PCR SSP, PCR SSO. These were the low resolution typings, the one that provided a first field typing. And sooner after that, the IMGT database was introduced in 1998, and this was because there was a need to have a reference database and to have a correct nomenclature. And not too long ago after that, there was more of a need to really identify the sequences more instead of only having low resolution typing. So Sanger sequencing was introduced, which is still looked upon as the golden standard of the high resolution sequencing techniques. So with the Sanger sequencing sequences in certain regions of the gene. Later on, with next generation sequencing, also not only the sequences were revealed very targeted, but the entire full-length sequences could be identified in a very high throughput nature. So to look a bit more at the evolution of the sequencing platforms. So as I already said, it started with the Sanger sequencing, which we consider the first generation platform of sequencing. Then the second generation was introduced in approximately the start of 2000. But a bit later on, it was quite later on that really people already started to using it in routine. So the next generation sequencing platforms, as we call them now, so the Illumina and the Ion Torrent platforms. And now we are starting to see an introduction as well of the third generation, which are the long read sequencing platforms. They're not really not very much used yet in routine labs because we're still struggling with a proper quality, but it's definitely coming and it's definitely the future. So what do we gain from NGS actually? What are the benefits? So one of the first benefits, of course, is the full-length gene sequencing. So as I already said, next generation sequences can amplify almost the whole gene of all loci. So this is the benefit. This comes actually along with the second benefit, which is the typing, that we can do a typing with zero ambiguities or almost zero ambiguities. And this gives one big benefit, namely no repeats or very few repeats, where with Sanger sequencing, you had to redo your runs with group-specific primers to resolve ambiguities. And with next generation sequencing, that's almost solved. Then another benefit of this is definitely the ability to identify novel and null alleles. This was already, you could already do this with the Sanger sequencing as well, but with NGS, it's more broader. It's in all exons that you can now identify the null alleles. Even you can look at the introns, and you can more easily also identify null alleles, which is also very important. Advantage of the next generation sequencing is definitely the flexibility in sample number. From low to high throughput labs can use this method. We have labs, customer labs, that do about 8 samples per run, but we also have labs as customers that do 288 samples in one run. So it fits actually to every lab and in every throughput. A fifth benefit is the possibility to have the pre and the post completely automated. So there's no need anymore to do any part manually. If you want, you can have everything automated, and to time in the meantime to do something else for your technicians. A last big benefit as well is the fact that it's very cost efficient, especially when you go towards higher numbers. The higher samples you do, the more samples you do, the more cost efficient it gets. And nowadays, it's not only for the high throughput labs, but it's also for the low throughput labs, because now we can, because it's also these labs that can combine their libraries together with other types of libraries. And that way, also for them, it makes it very cost efficient. So you can actually combine your HLA library with a library from the oncology department or pathology department. So you can then all benefit from it, and you can all lower your costs per sample. So to hit a bit more on the benefit from NGS regarding Sanger sequencing. So with Sanger sequencing, as I already said a bit, it targets more. It doesn't, I mean, you can, but it's too difficult to really identify all the exons in the intron. It's almost impossible. So Sanger sequencing, as you can see here, it was mainly for the class 1, the exon 2, 3, 4 that were identified. For class 2, it was mainly exon 2, 3, but that was mostly it, unidentified. And this gave actually a lot of ambiguities, because there were quite some labs that are starting to look outside as well. So NGS is different. So almost all loci are sequenced now full length. So meaning from 5' till 3' UTR, they are amplified. And all exons and all intronic information can be sequenced and can be identified. So this was already a big benefit, but there's also definitely an other benefit that comes with NGS. And this is the benefit of phasing. So phasing is one of these new definitions that came with NGS, because phasing is, let's say, it's simple. It's the separation of the alleles. So with Sanger sequencing, that was not really a possibility. So Sanger sequencing, when you did sequencing with your forward, your reverse primer, it gave you one signal, so to say. When you had a heterozygous typing, it would give you then a double peak at the heterozygous position. And in case you had two alleles that had multiple heterozygous positions, it was actually impossible to know which nucleotide would belong to which allele. So in this case, Sanger sequence would give you an ambiguity, and you would have to redo this run, you would have to redo this sample with group specific primers to be able to separate. You would have to reflux test. However, in next generation sequencing, this has been solved mostly because NGS actually directly gives you two signals. NGS can, based upon the heterozygous positions, can separate the alleles and directly give you a true heterozygous unambiguous result. And exactly how that is done using the sequence, I will show that a bit later. However, of course, next generation sequencing also comes along with quite some challenges as well, because it is quite new. And this for example, there are some new definitions, some new words like phasing, coverage, Q30 score. So these are some things that a lab needs to get familiar with. Then the algorithms, they're quite complex. NGS comes along with a high amount of data, and there are quite some complex algorithms that need to be developed in order to make sense of all this data and in order to analyze the good quality data. So this is definitely a challenge as well. Another challenge is the incomplete IMGT database that we are struggling with currently. The IMGT database has a lot of alleles, and the reference database is already very big. However, there are quite many alleles that are missing information, that are missing sequences. Especially for the HLA class 2 alleles, there's quite a lot of that do not only miss the intron information, but as well the exons. And this can be quite of an issue for proper phasing in our software. As a sort of contradiction, what is also a challenge is actually the increasing HLA database. Although it's great that people submit their alleles, and especially when they are completing missing sequences, it can be definitely a challenge. Especially the last years, we can see a very high increasing database. A lot of new alleles are submitted, and every time our software has to deal with it, with the analysis, with going through all this allele, but still having a short analysis time. So this can be quite a challenge as well. The biggest challenge when coming with NGS is the large and more complex data set. NGS, the NGS platforms give us thousands of sequencing reads from multiple samples at one time with multiple loci that can cross align to each other. And so you really need to have a proper software. The software is the key part here. The software is the thing that's crucial in order to make sense of the huge amount of sequencing data. And that is what I will be talking a bit later on. So let's first talk about sequencing platform, the sequencing platform that we are using. So our protocols can run mainly and actually only on the Illumina platforms at this moment. And our protocols are suitable for the iSeq, the MiniSeq, the MiSeq, and the NextSeq. And we've chosen their technology because it represents a low error rate and a quick sample preparation. So I think that most people are quite familiar already with the NGS, the chemistry behind the Illumina, but let me just recap a bit. So in our protocol, there's a library preparation, and this is important because you need to prepare your fragments, your library, to be able to sequence by the Illumina. So we're going to shorten our fragments. They need to have a certain size, and you're going to add indexes and the adapters. This is very important because with the Illumina adapters, your fragments can be bound to the flow cell in order for the bridge amplification to happen. So this bridge amplification is really important because every fragment will bind to the flow cell, and then with the bridge, through the bridge amplification, many copies will be made of this one fragment. So in the end, you can see here is the cluster formation, left bottom. And so you will end up with many, many copies, with many clusters, and every cluster contains thousands of copies of the same fragment. And these will then all be simultaneously sequenced. By every nucleotide that's built in, there will be a fluorescent signal. Depending on your cycle chemistry, whether you're using 300 cycle chemistry or 500, either 150 base pairs are sequenced or 250. In Illumina, you can either use single end read sequencing, you can do single end read sequencing, or pair end. But what is the difference? So on the left, you can see the single end. So with the single end, only one one fragment is only sequenced from one side. So only 150 base pairs from one side if you're doing 300. Pair end read sequencing is different though. Pair end means that one fragment will be sequenced from both sides. That's why you're seeing a read 1 and a read 2. So we can also say the read 1 is the forward and the read 2 is the reversed read. These Illumina sequences from the pair reads, so the Illumina knows that the sequences from the paired reads originate from the same fragments, and this enables the long distance phasing. I already said a bit about the advantage of NGS regarding phasing, so let me show you in a schematic way how we do that. So when Illumina platform creates this FASTQ files, because that is what those FASTQ files contain all the paired reads, our software will then, based upon these paired reads, will make a very long consensus sequence, and will try to say, okay, this sequence belongs to allele 1 and this sequence belongs to allele 2. So here you have an example of a heterozygous typing, a B07 with B41. And based on this pair end sequences, so the sequencing reads belonging to one and the same fragment, the software knows which nucleotides belong to one allele and which nucleotides belong to the second allele. This is what phasing is. The software automatically, based on these heterozygous positions that reside on one and the same fragment information, it can then separate these alleles. It can do the phasing, as we call it. And in this way, the software can directly give you an unambiguous heterozygous typing. This is in most cases. In very few cases, the software could still give you an ambiguity, and this is when the distance between two heterozygous positions is too long. Because our fragments are a certain size, they are maximum around 700, 800 base pair. If the distance is longer, that will give a problem. If the sequence is missing from the IMGT, that will be a problem as well. However, the troubleshooting behind this is something that my colleague Vera will mention, will explain and walk you through in the next webinar. So the pair end reads are very important, and these are analyzed within our analysis software, our HLA Twin software. So this is how our software looks like. So directly from here, you can analyze the data, and you can even do it in that way that it will automatically start analyzing once your MiSeq is finished. So just to hit up on the most important features of the software, it uses pre-configured holotype HLA specific settings. So these are thresholds, quality control metrics thresholds, specifically for holotype, and our omnitype, our multiplex as well. We have the automated genotyping after the MiSeq. So meaning, let's say that your MiSeq is finished in the middle of the night, it will then automatically trigger the genotyping in the software. The software uses up to 2 algorithms for determining the HLA genotypes. So these two are, first of all, the consensus genotyping algorithm, and the second is the statistical algorithm. The consensus is the main one, most important, because this one does a de novo assembly. It takes all the sequencing reads from the MiSeq, and then it makes one long contiguous consensus based upon all these sequences. It does not use the IMGT yet, it does it blindly. And then it goes to the IMGT to find the best match. So this one is very strong to find the novelty, to find novel alleles, to identify properly the full-length alleles. Statistical one, this is the one we use as a backup. By default static, it only will be triggered when there is a quality issue or novelty. Then another important feature in the software, this is something that our customers requested, because when you're in routine and you're checking results, you want to do it as quickly and as easy as possible. And next to that, you want to know when the software has good quality, so you can trust the data. So that's why we have introduced a traffic light system, and this is how it looks. So you have your HLA allele that the software is calling. And when you see these two traffic lights, then the first traffic light is the algorithm traffic light. This one, when it's green, then it says that the algorithms are fully concordant with each other up until the third field. However, if this traffic light is yellow, it means that they're concordant, but only up until the second field. If the traffic light is red, it means they're concordant, but only up until the first field. If it's red, it means they're completely discordant. They found something else. Then the second traffic light. This is actually the most important one. You will always see this traffic light. And this is the QC metrics traffic light. We have 24 QC metrics in our software. And when the algorithm starts analyzing everything, it will go through all these quality control metrics. When they all have been passed, then it's a green. When it's half green, it means they're passed, but there is some extra info. Maybe somewhere a threshold was just below or over the threshold, but it's still great. Then if the QC metric traffic light is yellow, it means you need to inspect. If it's half red, investigate, meaning go through the gene browser, check the sequences. If it's wet, then it means that it potentially failed. You would either have to rerun or maybe reanalyze with more reads. So we know that in about 95% cases, you will have great results. You will have a traffic light green or half green, showing that you can trust the data, you can trust the typing, and you do not need to further check anything or not need to do any troubleshooting. However, there will always be a certain percent of your cases that will need some further reviewing, some manual reviewing of you. And it's important that in these cases, you will have proper tools to troubleshoot, to know that you know what is going on, where should I check, and what should I decide. And for this, tools are very important. So let me guide you through the tools that we offer. The most important one that we offer is the quality control metric system. As I already said, we have 24 quality control metrics in the software. And these will guide you towards the cause of a quality issue and help you decide whether or not you need to repeat the run. So below you can see an example of one sample. You can see the typing, and below it you can see the table of the quality control metrics. We always have the primary QC metrics, which are the most important, secondary, and the metrics for troubleshooting. They are completely below. You cannot see it here right now. So when a sample is analyzed, our algorithm, our software will go through all these quality control metrics because the software wants to make sure that that it has the proper quality, the reads have the proper quality, and it can alert you when something is wrong. So let me zoom in a bit. Primary QC metrics and the secondary QC metrics. And so you can see that the primary QC are like the read count because you want to have enough amount of data, noise ratio, key exon spot noise ratio. This is very important because you want to make sure that your background signal is not too high. Then one of the main ones is as well the consensus coverage key exon minimum depth, which is marked, highlighted right here now. So the values that you see in the right mean that this is the lowest point of coverage in the key exon. So this is a very important one. Also, one of the final ones is the key exon allele imbalance. Because what you also want to know is whether the two alleles are balanced correctly, which is not, in some cases, is not always the case, but you want to know at least. We also have our secondary, as you can see below, which are, for example, the fragment size, read quality. If you want to know more about the QC metrics and you want to know the threshold and the ranges, you can just hover your mouse over the I button that you see at the end of a value. And then it will show you, as you see here, the criteria, the ranges of a passed, info, inspect, investigate, and failed. So you can check all of these. So in normal cases, when you have a great result, good quality, you do not even really have to go here because everything will be green and everything will be within the ranges. However, if there is an issue, then the software will alert this quality control metrics. And whatever quality control metric is alerted, then you know what you need to check, whether it's coverage or noise, or maybe fragment size. Fragment size is important because with a too short fragment size, you can get more ambiguities. So if this fragment size would have been read too short, you can then go to the data statistic tab where you can then see the fragment size distribution. I show you here one that has an ideal fragment size distribution. We wanted around the average around 400 base pair. As you can see, that is where the bulk is. However, sometimes you can get that maybe the fragmentation was not done successfully. Maybe it was not done on ice. Something went wrong. In that case, your fragment size distribution can be off, but you want to be alerted and you want to check. So below you can see an example of a fragmentation where there was an issue. And you can see now that the fragment size distribution is off. You can see that there are many short fragments at the beginning. You can see this peak of around 100 and even below 100 base pair. So this can be an issue, and then you know you would want to check more your sequences, your coverage, your typing. One other thing that is also important is to know, as I already said, allele imbalance. Are my alleles, are my loci balanced correctly? Ideally, you want that 50-50. You want, if you have a heterozygous typing, you want both of them to be equally present. If you have a homozygous, you want to be sure that it's really only one allele, it's really homozygous. In case, however, and this can happen in patient samples that have a low quality of DNA that are coming from a certain source, you can have, or in a certain disease state, you can have a sample that has a risk of a lower amplification of a certain allele. And you always want to be alerted by that. You don't want to make a mistake of reporting a false homozygous result. So here, for example, you see here you see an example of a very high allele imbalance within the key exon. As you can see, the second allele is at 1%. So the software is giving you, as you can see above already, a red traffic light. It's warning you, something is wrong. You check the quality control metrics, you can see extreme allele imbalance. Okay, so in this case, it's very clear. You can also check your gene browser and your sequences, but the software is very clear in here. Okay, there is still something. I cannot make a proper heterozygous typing, but there is something in the second. There is some second signal which you need to investigate. If you see this in quality metrics, you can also go to the data statistics. You can check also there whether you have an allele imbalance, because there you will always see the graph of the loci of a certain sample. And here it also shows that you have a certain allele imbalance. So this is always important, that at least there are some few cases that a software cannot give you proper heterozygous typing because the sequence of the second allele are too low, but at least it should warn you. And it should warn you that you should maybe rerun on with the NGS one more time, or you need to confirm it with another method, or maybe check a bit later when it's regarding to disease state. Another tool that is quite important to have, especially if you're doing 11 loci typing, is the use of the LD database, the linkage disequilibrium database. So in case you're doing 11 loci, meaning you're also typing HLA DRB3, DRB4, and DRB5, we all know that these loci are different from the others. They can be hemizygous, meaning that only one allele can be present, or that maybe even none of the alleles of a single loci can be present. So you can have that the data want to know, is the data missing because I made an error, a pipetting error, or is the data missing because it simply is not there, because the allele is not there, the gene is not there. So that's why we build in the use of the linkage disequilibrium database. You can check the linkage disequilibrium details table, as you can see it here, of that sample. And then in blue, it shows you your sequence, your typing based on your sequence. And in black, it shows you what is expected based on the information in the database. So based on this, you can see that your typing is what you would expect. So it gives you a more of a confirmation and security of your typing. Whenever you get alert in a QC metrics and a QC metric table, regarding to coverage, for example, or recount, you always want to check your coverage. And for this, you can go to the gene browser, and then there you can see your coverage plot, as you can see it here. You can see the coverage all across the gene, and you can see whether there is a coverage going down or not. Next to that, you can, on the above, you can click on visualization mode, as you can see. And then instead of just showing a plot, you can actually visualize the sequencing reads. As already said, we're doing pair end. So it always gives you a read 1 and a read 2, a forward read and a reversed read. Here you can see in pink the forward reads, and in yellow the reverse reads. So you can nicely see how the distribution is over the gene. You can even, if you would want to look more up close, you can click on collapse mode. Sorry, I went back. Click on collapse mode, and then you can even zoom in more. And when you zoom in with your mouse, you can go as deep as checking the sequences of the sequencing reads itself, as you can see here. This is something you definitely want to do in case you have a novelty alert. You would want to check the sequences of sequencing reads. Is the novelty correct? And I will show you now also how you can do that. So you would want to inspect your sequences in the gene browser. That is how you can use that. Something that is very important as well, I told you already about the noise ratio quality metric. When there is certain noise level, a background signal, you would want to check this, especially when the QC metric alerts you for it, that it's above a certain threshold. So in that case, you can go to the gene browser. You can click above on display noise, and this will include the noise track. This is how we call it. You see here on the left, the CN, consensus noise. This is the track where all the reads will be displayed that have been filtered out as noise. They are not used in the analysis. So here you can see an example where there is a noise, but it's very localized, it's very little. This is quite normal. You can have this in samples, that there is just few read noise that have been filtered out due to quality or cross mapping. This is quite fine. And the QC matrix will also not be very much alerted by it because it's still below the threshold. However, what happens with a contamination? How will you be alerted by that? So here you see an example of a contaminated sample, okay? And you can already see on the typings above, you can see the HLA A, B, C, DKB1, DRB1 typing. You can see a red traffic light. So this is already a very strong alert. Something is off. This is even, we consider, based on the QC metrics, this is even considered failed. You see this mark, this plus sign, it means that there's novelty. You can see that there's a novelty in all load size. Novelty. You can see a lot of times that there's a false novelty alert when you have bad quality data. So this already shows you, okay, something is off. Then you go to the quality control metrics. They are now shown below. And there you immediately see, and I highlighted it here in the red, you immediately see that the noise ratio is high, key exon spot noise ratio is high, and coverage, key exon minimum depth, is low. And this is an all load sign. So you can actually see that there is a high noise ratio in almost all load size. So it very clearly says there's a high background noise level of some sort. So you immediately know, okay, if this is the case, then I will need to go to my gene browser and I will check where this noise is located, the problem. So let's see. So then you go to the noise track. You activate the noise track in the gene browser. You see here the noise track in red marked. And you can see now, if you compare it to the previous one I showed you, here the noise is not localized. It's distributed all across the gene. You can see noise reads all across all exons and introns. This is a very good, clear example of a contaminated sample. So they actually confirmed that this DNA was contaminated, and that is why you saw contamination across all loci. So when you have this strong contamination, that the software has problems of giving you a typing because you saw that it had false novelties everywhere, well, then you know for sure you need to repeat this and that you will need to check where the contamination is located. And as final, I will talk about the tools that we provide to confirm a novelty, because this is something that labs always want to see, always want to know. If there is a sample with an allele with a novel sequence, how can I see that? How can I confirm that? So in case you get a novelty alert in the typing, then you can go directly to that position in the software as highlighted here. You can see that novel position are highlighted. So here, for example, you saw that the software has found the best match, the HLA 6603. However, there is one single position that is a mismatch, so that's different from the reference database. You can see here that the G nucleotide is what is in the reference database. However, our sequences have our sequencing reads contain a T nucleotide at that position. You can already see in the sequencing reads, you can see all T's. But obviously, you do not want to scroll down for all the sequencing reads. So that is why we have included the base statistics table. You just go on the position, right click, and it brings you to the base statistics. And here you can see the number of reads that contain a certain nucleotide at this specific position. So here we can see there are no reads that contain a G. So it seems to be for sure that this is a true novelty. We have good quality data, we have here a good coverage and they all contain T nucleotide. So once you confirmed that it's a true novelty, you then want to know whether it's a synonymous or non-synonymous substitution. So you want to know whether it changes the amino acid. For that, we have built in the display amino acid. You can see it on the top left. It's the fourth button from the left on the menu, display amino acid. And then it activates the amino acid track. That is how we call it. So you can see it here. You see first again the G and the T, the nucleotides. Now you can see that the G, as it was in the IMGT, encoded for glutamine. It's the blue amino acid, the blue box, glutamine. Our sequence, however, have a T. And this T nucleotide now encodes for the T within the codon, now encodes, it actually introduces a stop codon. It's not very good clear in the screen now because it's a quite bright green, but it says stop. So meaning that that this new T nucleotide now introduces stop codon within the exon 3. So based on this, you could actually already suggest that this is probably a null allele, and the lab that has identified this, they also already confirmed with serology that this is a true null allele, and they have submitted this already to the IMGT. So this is how you can check the gene browser, how you can check your sequence, confirm your novelty, whether it's true or false. And once you confirmed, you can check whether it changes the amino acid or not. So whether it will be a second or a third field nomenclature change. So in summary, I've shown you that NGS has many benefits over previous HLA typing methods, like almost zero ambiguities because of the high resolution, and also much more cost efficient. However, there are also quite some challenges, and one of the biggest challenges that NGS gives you a very large and complex amount of sequencing data, and that the software is one of the most crucial parts of the whole workflow to make sense of this data. And then I showed you that it's crucial and it's very important to have the correct tools. So I showed you tools in the software like the quality control metrics to alert the user for a potential issue, to guide you towards that issue, and to help you make a decision. I showed you the gene browser to manually check your sequences, to check the coverage, and to potentially confirm a novelty. I showed you the noise track within the gene browser to check your potential contamination, to check your noise level. And I showed you the linkage equilibrium details table that uses the LD database to confirm, to give you more security for your more confidence in your 11 low side typing. So with this, I want to thank you for your attention, and please let me know if you have any question. Put them in the question box, and I will try to answer them as best as I can. In the meantime, I want to tell you that everyone will receive a follow-up email with the recordings after this. In case you need the HI credits, then please fill in the survey. So you will only need to fill in the survey if you need these credits. For all the rest, you can already get the recording if you would like. So let me check if there are any questions. So for now, I have not received any questions. Okay, I have received one. How long does it so okay, so there's one question and it says, how long does the typing take? Okay, so this is more regarding the work. It's a good question actually. So at the moment, so we have our singleplex. Okay, let me check. Yeah, so we have our singleplex protocol and we have our multiplex. The singleplex is a 1.5 day workflow and the multiplex is 1 day. So we are releasing right now, we will release the multiplex 11 load site kit. And this means that within 1 day, so you start in the morning with your DNA, and at the end of the same day, in the afternoon, you can start your Illumina platform, let's just say your MySeq. So the next day, the sequencing will be finished. So you can start analyzing and having your typings. So within the second day, you can have your sequences, your typings. Let me check. I have another question. Is the novel allele info exported along with the typing data? Yes. So we have an export. So we have in the gene browser, we have an export feature. You can export your data. It exports the sequences into a FASTA file, and it also export the information that you need regarding that allele for the allele submission. Yeah. Let me see, I have another one. Does your software work only for your kits? Okay, so the question is if our software only specifically works with our holotype HLA workflow with our kits. Actually, no. So the HLA Twin software can handle other data. As long as it's FASTQ file, it can handle it. However, the HLA Twin software is provided together with the holotype and with our OmniType multiplex, which is coming out. So normally we do not provide this one separately. However, we have a software which is called Explore, and this is separately. You can order this software on its own, and this is really meant for people which only want to use the software a lot of time for research lab, which have data from different kind of sources, and it can also handle whole genome data, for example. So this is the Explore software, and it works in the same way actually as the Twin software, but this one you can. But normally the Omexen Twin, the one I showed you now, this comes along with our holotype kits automatically, and you also do not have to pay for it separately. Once you order it, you get also licenses for the software, as many as you like. Okay, let me see another question. How do we know the exact location in the gene browser to be checked when the software suggests multiple typing? Noted. How do we know the exact location in the gene browser to be checked? Sorry, multiple typing. So is this, so I guess you mean when you get a potential novelty, maybe? So the software actually has a feature that shows you in the software, you can actually see it has a button, show novelties, and it directly shows you the specific novelty at the specific specification. You do not need to search for it in the gene browser. You do not need to search for it in the gene browser. The software will lead you to it. Yeah, so it always has a novelty and alert, and it always leads you to it. So this is in case when you have a novelty alert. And in case you have heterozygous typings, it will always automatically show you the heterozygous typing in the gene browser. So it always automatically shows you the 2 alleles and the sequences of the 2 alleles. Okay, I have one other question. Is your software cloud-based, or do I wait? Is your software, or do I need to install it on my computer? So it's both. It can be installed in cloud. It can be installed in the software. It can be installed in the server. It's all up to you. So it's very flexible when it comes to that. I have customers that have it on cloud-based, I have customers that have it server-based, even customers that have high throughput but use a very good computer for it. So it's totally up to you. And we have our specific IT support that can help you with that, and that help you to go through the installation process. Is there any other question? I think that was it. Please let me know if you have any other question, or else I'm going to close. I also want to leave this, and I want to just state that in the next webinar, which will also be very interesting because my colleague Vera Sifranova will be presenting some very nice troubleshooting cases. So this was an overall view of the tools that we have in the software, and she will show you really how to go through certain difficult samples. For example, a cis-trans ambiguity or novelty. So that will be also very interesting. So please join us as well for this one. And I don't think there's any new questions anymore. So then I would like to close the session, but please, if you have any other questions, just email me on the email that is in the screen, and I will answer to you as soon as I can. Thank you again for your attention, and have a great day.